many different types of microorganisms have developed ways or strategies for evading the complement cascade because it's so detrimental to them. Many of your more important and successful pathogens have some sort of strategy for doing that. There's a good section at the back of this chapter that talks about that, and it talks about some of the strategies that are employed, and it also has a great kind of advanced box that they're talking about Staphylococcus aureus and all of the different ways that they can avoid destruction by the complement cascade system. So the complement cascade has probably pushed the evolution of some of the pathogens in certain directions. So if you look on page 216 in the 7th edition, there's a box called Staphylococcus aureus employs diverse methods to evade destruction by the complement cascade. I mean, some of the bacteria have evolved very simple techniques. They have a really thick peptidoglycan, so they just kind of slough off some of the activity of the complement cascade proteins. The gram-negative bacteria will have a shield of magnesium ions in their outer membranes that help to repel some of the individual complement complex proteins. Forming a thick capsule means that the activation occurs on the very outer edges of this slime coat, and they're constantly kind of sloughing off the outer layer of that slime coat. So the complement cascade never gets close enough to the cells. Then they have some more specific things that they can do, and different organisms use different techniques. I'm just going to talk generally and let you read the chapter. Okay, some pathogens can interfere with the first step of immunoglobulin complement activation. So they can do that by binding specifically to the FC regions that would activate complement fragmentation. There's also some that produce microbial proteins that inactivate complement proteins. So they'll produce proteins that bind to specific regions on the complement proteins and prevent formation of, say, C2A 4B. There's some parasites that do this as well. There's schistosomiasis, Schistosoma japonicum is a big one, and Trypanosoma, both cruzi and brucei. Both the one that evolved in Africa and the one in South America have the ability to produce proteins that will bind the complement C2 receptor protein and disrupt the interaction between C2A and C4B, and thereby they prevent the C3 convertase from forming. There's also some proteases that are produced that destroy complement proteins. So complement proteins are produced, and there's an aspergillus that produces an alkaline protease that actually cleaves several of these complement proteins that are there. Some microbes have learned to mimic complement regulatory proteins. So they've, especially when you're talking about viruses, they have, in some cases, incorporated the gene for the regulatory proteins like tumor necrosis factor or I'm just completely blank. The factor H, the decay accelerating factor, not tumor necrosis factor, and the S proteins and things like that have been found in viruses. It's a little off topic, but some viruses have actually picked up genes to generate complement proteins because complement proteins can have an autolytic effect or an autocrine effect, allowing the cells to go through faster cell division. So they're excreting certain types of cytokines. So it's really an interesting area of research, and they're still learning more and more about it every day. But I need you to read through that chapter and be able to give me an example or two of the way that some microbes have evolved strategies for evading the complement cascade.